Hello. In this video, I'll be presenting on the Module 12 journal article, Label-Free Microarray Imaging for Direct Detection of DNA Hybridization and Single Nucleotide Mismatch by Kumar et al., published in 2009, which discusses an alternative method of detection of DNA hybridization on microarrays. A DNA microarray, or DNA chip, is a common tool used in biology and medicine to analyze DNA structure based on specific binding energies. DNA chips are extremely helpful in analyzing hundreds of thousands of genes very quickly and with a very small sample size. In a typical DNA chip setup, a known DNA fragment is immobilized in different wells of the microarray and analyzed via hybridization of the known DNA with the DNA of an expressed gene. To use these DNA microarrays, genes extracted from the experimental sample are fluorescently tagged with one color, in this case green, and a standard sample is tagged with another color, or in this picture, red. These tagged samples are then added to the pre-prepared DNA microarray so that hybridization can occur. Samples that bind with the experimental sample will fluoresce green, and those that do not will fluoresce red. The amount of samples that fluoresce individual colors is then analyzed. One of the ideal functionalities in DNA microarrays is their ability to detect single nucleotide polymorphisms, or a single difference in the Watson-Crick base pairing. Unfortunately, with the current microarray and fluorescence detection methods, it's incredibly difficult to detect the small differences in binding energies between the single mismatched and the perfect hybrids. In addition, the process to do so is incredibly difficult and the results are hard to reproduce. Therefore, the problem that this paper aims to tackle is, how do we perform high-throughput detections of single nucleotide polymorphisms, or SNPs, in DNA? Luckily, label-free methods have been proposed as a possible technique to identifying SNPs without the need for fluorescence labeling. Specifically, this lab has recently developed their own spectral reflectance imaging biosensor technique, or SRIB. In this method, a microarray is placed on top of a silicone oxide reflector. This layer is hit by a laser with different wavelengths of light, and the light reflected off this layer is passed through a beam splitter and into a photodetector, which measures the thickness of the layer and any interference due to the biological material in the microelectrode array. For this study, the researchers chose two DNA lengths, 18 and 20 nucleotide long strands. Each of the DNA types that these researchers used were duplicated for a total of 40 spots for each DNA sample type to be analyzed. The figure in the middle shows the organization of the different DNA strands, where PM stands for perfect match, MM stands for single mismatch, and DM stands for double mismatch. The last group, or the 40 negative, is a random DNA sequence and was used as a control group. The graph on the top right shows the results of these wells being analyzed with the SRIB technique for each group, both before and after hybridization, shown in black and red respectively. And the bottom graph shows the average percent change in mass after hybridization. As you can see, this detection method was able to differentiate between the double mismatch and perfect match groups very well, as demonstrated by the height of the mass bars, but was not able to sort out the single mismatches from the perfect matches very well, as seen by the height of the mass bars not being statistically different. In the next part of the study, the samples that did experience hybridization were washed with various salt concentrations to determine the kinetic measurements of denaturation, or splitting of the two strands of DNA. As salt concentrations for washing the samples are lowered, the charge on the DNA backbone is reduced, which causes the DNA strands to denature and decrease in mass. The figure above shows how the different DNA samples denatured at various concentrations of salt. Since almost all of these groups dropped drastically at the 0.625 millimolar level, it was determined by the researchers that the mass change was not a sufficient measurement alone of the stability of the DNA or the ability to differentiate between unstable double mismatched strands and stable perfectly matched strands. From the decay curves in the previous slide, the 20 perfectly matched, single mismatched, and double mismatched samples were pulled out to generate the graph on the left and fit to an exponential with the equation F shown here for all times after the 0.625 millimolar concentration onset time. The results of the curve fitting can be seen in the small box on the left figure. The main parameter of interest in this situation is A2, or the decay constant, which demonstrates how quickly the sample denatures. These decay constants were plotted for comparison in the graph on the right. As you can see, the perfect mismatched and single mismatched constants are distinctly different for both the 20 and 18 nucleotide long strands. When comparing the 20 and 18 perfectly matched strands in the bottom left of this figure, you can see that the decay constants are also different, indicating that the 18 long strands are less stable than the 20 nucleotide long strands. This demonstrates that perfect mismatched and double mismatched and single mismatch could be distinctly differentiated from each other using this curve fitting tool. In conclusion, the SRIB system can perform high throughput screenings of DNA and detect hybridization of DNA strands easily via the change in mass of each microarray well. Comparison of the masses, both before and after hybridization, were deemed by the researchers to not be the best way to determine single and double mismatches from perfect matches. And finally, kinetic measurements of denaturation can be used in conjunction with the SRIB technique to differentiate single and double mismatches and perfect matches from one another.